Hey, my name is Justin Lowe. Thanks for joining me today at Entomology 2021, and thanks for choosing to come to my talk. Uh, on behalf of all my coworkers, I'm presenting our work today called Ladybeetle Community Composition is influenced by local scale restoration, but abundance is affected by seasonality. This work was undertaken by mainly employees and myself as a research affiliate at the Cheadle Center for Biodiversity Ecological Restoration at UC Santa Barbara, and I'm currently a PhD student at the University of California, Santa Cruz in the Environmental Studies Department. If you have any questions about this talk later on, feel free to email me at this email here, or you can reach out to me on my Twitter at Justin C. Lowe. Before we get started today, I just want to take a quick moment to do some acknowledgements, including a land acknowledgement stating that the work that was undertaken here was on the lands of the unceded territory of the ancestral Chumash people who were forcibly removed from their lands and today are working to heal and decolonize from historical trauma. Our funding sources, the Cheadle Center and the UCSB Coastal Fund, and also I wanted to thank our many numerous undergraduate research assistants that helped in lady beetle surveys. Native lady beetle populations have been declining since the 1980s, and some have estimated certain populations may have been reduced up to 60%. Declines are caused by many reasons, but one of the leading thoughts and theories is due to habitat loss, and that some of the ideal habitats for native lady beetles have been transformed through urbanization or for agriculture. Native lady beetles are also declining due to competition from invasive and non-native lady beetles, such as the seven spot pictured here on this slide, and also the multicolored Asian lady beetle. These species are both very competitive and can often be more voracious predators than their native counterparts, competitively excluding them, and also directly impact native populations through intragill predation, where invasive or non-native ones can also consume and directly reduce native populations. Ecological restoration is often used to help offset the loss of biodiversity. Ecological restoration is often undertaken through planting native species and removing non-native plants. Oftentimes, we expect that if we restore a habitat, native wildlife will return naturally, which in the literature is often referred to as the field of dreams hypothesis. However, more recent work often has found that the field of dream hypothesis does not necessarily hold true for all taxa and often is less prevalent for higher trophic level species, such as coccinellids or lady beetles. In order to better understand how coccinellids are responding to restoration, both native and non-native, we asked the following research questions. How does land status, such as restoration and remnant status, affect native and non-native lady beetle abundance and richness? If we restore these habitats, will native lady beetles naturally recolonize? Does local scale restoration affect community composition of coccinellids? And how does seasonality influence native and non-native coccinellid abundance? In order to address some of these questions, we used a study design that involved surveying remnant, restored, and non-restored grasslands in Santa Barbara, California. We set out to survey 3 of each of these sites, and we undertook collections from 2017 to 2019. During the years that we did lady beetle surveys, we did weekly collections, or often collections twice a week, at every single site for 10 weeks of every season during the year. Season was defined by the solstice and equinox calendar dates, and oftentimes we used visual surveys in teams of 2 and captured the lady beetles whenever possible using multiple methods such as sweep netting, aerial netting, aspiration, and hand collections. We also noted the associated plants, native or non-native, that each coccinella that we observed or collected was interacting with. So this is a summary of our results, and on the figure on the right, you can see a figure on the x-axis is the season, and on the y-axis is the log transformed native or non-native coccinellid count. And then as the bar graph indicates, you can see that Red is from non-restored sites, purple is from remnant sites, and green is from restored sites. We found that native and non-native coccinellid abundance was primarily affected by seasonality and unaffected by the land status that the site was, that the lady beetles were collected from. We also found that native coccinellids were more abundant in the spring, and non-native coccinellids were also abundant in the spring, but they had low populations in the summer. We found that the richness of both native and non-native lady beetles were unaffected by both land status and by seasonality. We did see that restoration affected coccinellid community composition. Restored and non-restored sites had different community compositions, as you can see in this non-metric multidimensional space plot, but they were both similar to remnant sites. So let's just take a moment, also now just take a moment to look at the MMDS, and I'll explain a little bit about how it works. So the points represent the actual sites that we collected the lady beetles from, and the shaded in polygon represents this non-dimensional space that the community encompasses. When there's overlap in these shaded in polygons, that means that these different treatments are similar. If there is no overlap and these shaded polygons are separate from each other, that means that these communities are distinct and that the treatments may have affected the community composition, which is the case for restored and non-restored. We also found that remnant sites had the most specialists and that restored sites often lacked the specialists that remnant and non-restored sites had, which were Coccicoidea, scale specialists, and Eristophiles, which were lady beetles that specialize on mildew. To summarize, we found that in our 2-year study that restoration did not affect or increase the abundance of native coccinellids, nor the abundance of non-native coccinellids. We also found that restoration lacks specialists and that oftentimes it can even have less specialists than non-restored sites. And this may be due to the amount of disturbance that restored sites receive because they're actively managed with planting and weeding. And in the future, with reduced maintenance, we may expect that specialists could return to these restored sites. We also found that seasonality impacts coccinellid abundance and had a greater varying effect on abundance than restoration or land status. And we didn't find anything that strongly affected the richness of the coccinellid communities. And to take in mind that this is also done with highly intensive sampling taken almost every week, 40 weeks out of the year. Our results indicate that restoration management may need to incorporate strategies for reintroducing or improving native wildlife populations, because contrary to the Field of Dreams hypothesis, we did not find that native coccinellids naturally returned to restored sites after they were restored, countering the notion that if we build it, they will come, or if we restore it, they'll return. This means that restoration may need to consider additional facets of diversity beyond paleont taxa to restore higher trophic levels, and think potentially more about functional diversity and potentially phylogenetic diversity, and potentially whether or not those in the future could aid in improving and informing restoration of higher trophic levels. So I want to thank you all again today for electing to come to my talk and making it this far. If you have any questions at all, feel free to reach out to me at my email on the slide here, jlung4@ucsc.edu, or if you just want to follow my research updates, you can also connect with me on Twitter at Justin C. Lung. Thank you again. I hope you enjoy Entomology 2021 and have a great rest of your day. Thank you.